good afternoon. I am presenter, myself Shwetakshi, and I will be presenting on Germination Test for crops. So before moving on, we have to first know what is germination. So by definition, germination is the process by which a plant grows from a seed. In this process, there is emergence of radicle and primule through the seed pod. So as you can see here in the figure, there is a seed and it gradually grows to form a plant. So the entire process of a seed being formed to a plant is called germination. This is a very important part. At least it is the key, because if there is no germination, there is no plant which is going to be formed. So it is very important to know how exactly we should germinate the seeds.
Now there are certain requirements for germination. They may be broadly categorized as external factors and internal factors. Now the external factors include adequate amount of water, a good temperature, proper aeration, and exposure to light and dark. Whereas the internal factors includes the seed viability. That means how much is the seed potential to be formed into a new plant. Then its genotype, seed maturation, and seed dormancy. Now there are three different stages involved in seed germination. They may be classified as pre-germination, germination, and the post-germination. Now suppose when you are sprouting moong, that time what we do first, we take the moong seeds and put it in water. So the seeds of moong gradually uptake the water, which is called as imbibition, and all the biological process gets activated inside the seed. So it is increased metabolism, which results in the changes in the shape of the seed. So all these steps comes under pre-germination step. The next one is the germination, where actual rupture of the seed coat occurs with the emergence of the radicals. So as you can see here in the figure, there are small radicals which are being coming out of the seed. So this is the actual germination. And the post-germination, which include the growth of the radicle and the emergence and development of primule. So now we will move on to the germination test. So germination tests are certain procedures that will help us to predict whether the seed has enough potential to germinate and form a new plant. Now there are several significance of doing this germination test. It is used for sowing purposes. So if you are sowing seeds in a garden or in a field, you should know whether the seed which we are sowing is potential enough to germinate into a plant or not. And it is also used for labeling purposes, seed certification purposes. So different industries which are involved in selling seeds, they should give a certificate that yes, the seeds which we are providing are going to give you a better yield. And this is done by using the germination test. Other significance include the experiment purposes, the general home purposes, and the Seed Act and law enforcement purposes. So even before going to the seed germination test, the simple procedure to select the seed which could be good, that means to predict, is using a glass of water and inserting the seeds inside it. So when I submerge the seed inside a water of glass, so the seeds which sink are actually good and the ones which float are not appropriate. This is because when we submerge the seeds into water for say 10 minutes, the seeds will start absorbing waters, and those seeds which are good and have a viable embryo, they will sink because the density is increased, whereas those which float may not be that much having that potential to be grown into a plant. So this is one of the traditional technique of looking at the seed selection. Now there are different germination tests, but the common ones are the roll towel method, Petri plate method, sand method, and the between paper method. So we will be discussing about each of them. The first one is the roll towel method. So this is a method which even I tried at my home. You can see here, first we need to take some seeds and roll it using a tissue paper and then cover it with a plastic box, or you may even use a Ziploc bag. Next step is to wait for 2 to 3 days, and then you can observe that small radicals have started coming out of the seed. Then gradually these radicals will keep on increasing in their size, and in my experiment at day 5, you can even observe that the even smaller radicals have coming out from the main radical root. So this is becoming like a root system. And simultaneously, there will be a growth of the plumule taking place. So by day 6 here in the experiment, there will be small leaves which have came out of the seed. And I did this experiment for 7 days. So you can see that by the end of the 7th day, I could see a radicle, which is the root system, and the plumule, which was the shoot part of the plant, have emerged. So this is a roll towel method. Another method we can use is the Petri plate method. So this method is ideal if you're using small seeds, say mustard. So if you're using mustard seeds, we cannot, it is really very difficult to use the roll towel test for this because these are very small. So we can use a Petri plate method in which we will take a Petri plate and cover it with a wet tissue or a wet cloth and place the seeds over it. Now, this is also good if you are using seeds which require sunlight because you can directly put the Petri plate near the sunlight and this will be beneficial. And it is a common and easy procedure in the lab and home. Another method used is the sand method. So sand method is ideal if you are dealing with bigger size of seeds. So as you can see here in the figure, there is a tray and we need to add a uniform layer of sand through it and make small holes at equal distances and insert the seed inside it. And then we can cover it again with a layer of sand of say 1 to 2 centimeters in depth. So after 4 to 5 days, you can then observe the germination of the seeds. And the last one is the between paper method. So it does not require an extra container. So you can see here in the previous slides, this was a roll towel method which required the extra container or a plastic bag to carry it. Then Petri plate method which require Petri plates. And then the sand method which also requires a lot of sand and tray. But this method is quite easy. You just need to take a paper and fold it like a Japanese fan and insert the seed inside this, as you can see here in the figure. So the seeds could be of varying sizes. It does not matter here. And even you can grow it to a much taller height as compared to others. So this is the between paper method. Now once these seeds are germinated, we must know the germination percentage of the seeds. And for this, the simplest criteria could be just label the number of seeds and say whether they are germinated or not. So germination, that means whether the radicle and plumule have emerged out of the seed or not. So for my experiment, I took 10 seeds and you can see 9 of them got germinated and 1 was not germinated. So the germination percentage for my experiment will be the number of seeds germinated upon the total number of seeds into 100%. So 9 upon 10 into 100%, which give me 90% germination. Usually, if you are getting 70% and above, it is acceptable. And even you can add many different parameters to this table, like the time of germination, the length of the radicle, the length of the plumule, how gradually they have elongated. So they can be even more complex parameters, but the simplest one would be doing it this fashion. Now, what to do once these seeds have been germinated? Either you can plant them, so it can be used for the plantation purposes, as well as we can even consume them as sprouts or in some salads. So they are very nutritious because all the bio compounds are in activated state at this time. So if they are activated, it can add to your nutritionist values. So that's all. Thank you.
Thank you, Shwetakshi. It was a wonderful presentation made by you. I hope everyone is really got the information about how to go for a seed testing or to know what is the seed percentage for their quality seeds they have bought from the market. So now we have a third speaker, Mr. Daniel D'Souza.